starting on slide 28, structures of the heart. So we have gone over a lot about the heart already. You've had the heart in lab, basically 2 weeks, dissection and your practical. And now we're repeating all of that. Most of that is not new. There is some new information that will be on your exam, in addition to the entire endocrine chapter. So just reminding you. And there is we're just not going to have homework. There's no homework due on Friday. None at all. Okay? And I have heard happy things about the extra credit quiz that most of you decided to do. I've heard that grades went up. So that's exciting. And your previous quiz, I did go back and curve it slightly because it was out of 20 and the top score was 18. So I think I curved it to 17. Yeah. So that should hopefully help you as well. So I'm encouraged. Seeing grades going up is always good. So be encouraged about that. Break is just a couple days away, but don't go spring break in your head yet. Wait till after Friday's classes are over. I know, try to focus, just try. Just as a reminder, you have 2 weeks till your next lab. Remember, you do have coloring due in lab, next lab period, and you'll have your next muscle quiz. Those of you who have Wednesday lab, you'll find out about that today. Okay, continuing on with the lecture. Apologize. Okay, so we've talked all about the internal structures of the heart. So we're going to look at external things just a little bit. And in this picture, you can see the major blood vessels. And we've already talked about, we have 4 sets of vessels, 4 chambers, and 4 valves. Okay, so looking at blood vessels, okay, we have those 4 sets of vessels. See, pulmonary trunk. Oh, seriously, draw. Where's my color? Where's my color? I can't draw. Hold on one sec. Am I in the wrong place? There we go. There we go. Okay, so the pulmonary trunk leaves which ventricle? Yeah, what ventricle is the pulmonary trunk attached to? The right. Excellent. And so that is going to carry blood to the lungs. So you have your left. It's not showing the right pulmonary. It just has to be plugged in super tight. Okay, so your pulmonary trunk branches to your left and right pulmonary artery. So that's a set. And then blood coming back from the lungs travels in what set of vessels? Blood from the lungs comes back to the heart in what vessels? The pulmonary veins. Excellent. And so that's going to dump blood air into what cavity? Your left atrium first, then your ventricle. Okay. So just remember those valves and the vessels. And so then the aorta is also a pretty important one, and the superior and inferior vena cava as well. Okay. Just remembering that the point of the heart, that is called the apex. So make sure you're familiar with that term. That might pop up. I can point to the apex on a picture of the heart cut in half. So make sure you remember the apex is the tip of the heart. You know the auricles. We've talked about that. Okay, we're going to move it on. Okay, coronary arteries. And in lab, you identified an entrance to a coronary artery. Do you remember where you found that entrance? It's from the heart. It's from the lab. So where is the entrance to a coronary artery? We identified that in lab. Jeffrey? Yeah. So it's like at the very beginning of the aorta, behind one of those aortic semilunar valves. And so blood shines straight into the coronary artery from there, and there are other coronary arteries. And so the coronary arteries are getting highly oxygenated blood, and it travels through the heart muscle itself, supplying the muscle like we've talked about. Veins collect that deoxygenated blood and dumps it into what structure in the heart? Where is the oxygen poor blood collected? Say it louder. The coronary sinus. Remember that? And then remember it now. The coronary sinus has all the oxygen poor blood. It's collecting from the heart muscle itself, and then it dumps that oxygen-poor blood into the right atrium. Okay, so it's recycling it. So the whole point of having coronary arteries is, again, to supply oxygenated blood and nutrients as well, so you can make energy, to the myocardium, the muscle of the heart itself. And the reason we need to have vessels is because the muscle is too thick for the nutrients and gases to diffuse through the walls of the chambers to the muscle. It's just too thick. It can't do it. And so the whole heart itself has a ton of capillaries branched out for gas exchange and nutrient exchange. And so we looked at one of the entrances to the coronary arteries. There's actually two in the aorta right behind those semilunar valves, but we just looked at one. So there's a right and a left coronary artery. And so the term coronary refers to a crown, and I'll show you a picture in a minute that kind of illustrates why they were termed coronary arteries and veins. Okay. So here is a picture, and it's just illustrating the arterial system of the heart itself, and everything else is just kind of in the background, and we just want to look at the coronary arteries and the branches off of those. So remember, we have a right coronary artery that's branching off to the right side, and a left coronary artery branching off to the left side. And if we look at this picture, we can see that it does wrap around the back of the heart, and it wraps around the back as well. It looks a lot like a crown, which is kind of neat. So the fact that they kind of look like a crown surrounding the heart is where we get that coronary name. And so let's see here. We already know that blood enters those coronary arteries in the aorta. Let's see here. So when you looked behind the aortic semilunar valve and we saw that little hole, the opening to one of the coronary arteries, it's kind of hard now. We don't have a heart in front of us to double check this, but behind the pouches where there is an entrance to the coronary artery, it's a deeper pouch. So they call that the aortic sinuses. So you have these deeper pouches, so blood easily collects there and then shunts it into a coronary artery. Okay. Most of the stuff you guys are very familiar with already. Okay, so then deoxygenated blood, oh good, I have blue, which is perfect. So deoxygenated blood is going to return, collect in our coronary sinus. So this is a posterior view of the heart. So just as a reminder, this back portion here, this area I'm trying to color in with blue, this is the coronary sinus. And we're looking at, again, the posterior view. So this is the right side of the heart, and this is the left side. And so right here, this is the right atrium. So it's going to dump that blood right into the right atrium, and then it's going to be recycled, which is really cool. And we did look at this already. We've seen it in lab. Okay, now here's an interesting idea, skeleton of the heart. And I'm going to pull this up online on the big computer so we can see the animation. So bear with me for just a second if we can get YouTube up and going. Okay, so as you guys saw, most of the heart is muscle. We've seen the valves. Valves, what color were the valves? Lighter? Yeah, they were kind of like light pinkish color. The chordae tendineae, the heartstrings, they were white. And usually when we have like a white tissue or a tough tissue, it's some sort of connective tissue. And so that brings us to the whole idea that the heart itself has a skeleton, which sounds weird, but it's a connective tissue skeleton, and it helps give the heart its structure, and it's very interesting. And it says the 4 valves, so we have our 2 semilunar and our 2 atrioventricular valves. If you lined them up just right on the heart, they're all in the same plane. So if we did a transverse section of the heart, we would be able to see all 4 valves if we just did it right in the same spot. And that's kind of neat. And so if you notice here, it's in highlighted, the connective tissue of the valves is united within the center of the heart. We can't see it because we don't do a dissection within that one little spot that it's talking about, but it is all interconnected. And let's see. And one interesting thing about having this skeleton, this connective tissue skeleton of the heart, is that the right side and the left side, and like the atriums and the ventricles, they're not going to directly stimulate another part of the heart to contract. So it helps, because you have this connective tissue barrier essentially, it helps make sure that the contraction cycle happens exactly as it is supposed to. Give me 1 second. Let's see if I can just type this into YouTube real quick. I know you can't see what I'm typing. I'll switch it over in a second. Maybe it's not going to do it. Thank you for your patience. 1 more second. I'm going to go to the other file. Or we could not watch the video. I mean, the video's pretty fun. Okay, there's our link. Oh. My bad. It's not going to work. Let me try one other thing, see if we can at least get you a better picture. All right, so I typed into Google connective tissue of the heart. I'm going to switch over to the computer real quick so you can see this. There we go. All right, so first of all, do you remember the very beginning of the year we watched a video about how they are able to create hearts with your own tissue, your own cells on it? They washed away all the muscle cells and all you had left was connective tissue. That, first of all, is a really good example of the connective tissue skeleton of the heart. And so this picture here is just showing you the heart without the heart cells, just the connective tissue. And so it does preserve the shape really wonderfully, but most important, I want to show you how those valves are lined up. If they have a quick, easy picture here, possibly this one. Let's see. So this one, this heart is injected and preserved, but if we look here, we have our atrioventricular valves. Look, notice how they're right across from each other. Right here is your pulmonary semilunar valve, and right behind it is your aortic semilunar valve. So this is a good illustration of how they are within the same plane. And notice, I mean, we have walls that separate all 4 chambers independently from each other, and that is important in how the heart is going to contract and function. Okay, going back to the slides. Okay, so this important fact here that, yes, again, one contraction is not going to directly stimulate another contraction because you have that separation of all the chambers. Okay, so talking about how the heart contracts, how this works. Okay, so the conduction system, how the signal is started for the contraction of the heart, how does it spread, how does the heart contract. So that's what we're going to be talking about now. And so we say that the heart is autorhythmic, which means that, so auto means self, that it is controlling its own rhythm. Okay, so the nervous system is not directly controlling the rhythm of the heart, which is kind of like the lub-dub sound when you're listening to it through the stethoscope. So yes, it generates its own rhythm when it's beating. Your brain can speed it up or slow it down, but it can't alter the actual rhythm of something. Okay? And so notice here it says the rhythm is dictated by a series of structures, specifically cellular activities of the heart. So there's a group of cells that you find in the right atrium, and we call that the sinoatrial node or the SA node. And this group of cells, yes, I did say yes, up in the right atrium, you can't visually tell the difference between those cells and any other cells, but they have their own rhythm. If you looked at these guys under like a dissection microscope or a regular microscope taken from a live heart, you can see the little cells contracting. They have their own little rhythm going on. And so the rhythm, the contraction cycle, is started at the SA node, which is really neat. And so in this little picture, in this little picture here, it's showing the SA node is this lovely yellow dot. And again, remember, you can't visually see it. It's just a group of cells that are generating this rhythmic beating. And what's interesting is if one group of cells that were the sinoatrial node stop functioning, the next group of cells that they're all autorhythmic will take over. Okay. So, continuing on, as I mentioned, every muscle cell is autorhythmic, but the SA node has the cells that are more, I don't know how to describe it. Oh, right here, sorry. They have the most rapid rhythm, are going to take control of the rhythm of the heart. And so, as I mentioned, if those somehow stop functioning, the next fastest set of cells will become the SA node. Okay, so remembering every muscle is autorhythmic and interconnected. The way they're interconnected, if we remember and we back up to when we were talking about cellular junctions, they're interconnected. First of all, heart muscle is branched and they connect via those intercalated discs. Remember that? Some of the special characteristics of the heart. And those intercalated discs, you have gap junctions where the cytosol, the fluid from one cell, can spread to the next cell. And in this case, if you're trying to spread the signal for contraction, ions are passing from one cell to the next, triggering the contraction as it moves down. Okay? So remember, all cells receive signals from the more rapid cells before they can generate their own, so they follow the most rapid. They always follow the most rapid. Okay? And so remember again, this is your SA node, and it's very commonly referred to as your pacemaker of the heart because, okay, if we just take the word apart, it is controlling the pace, the rhythm of that contraction cycle. So it's really cool that it works. And so, again, reiterating what I said, if the current SA node fails, then the next set or the next fastest or rapid muscle cells will take over and be the next SA node. So the next fastest will always take over. So just remember that. So you want to, let's see, wait, did I say that wrong? I'm double checking. Okay, so apparently I did say that wrong and I apologize. So I'm going to read this last part to make sure we don't remember the wrong thing. So if those group of cells that are the current SA node fails, it says other less rapid areas could take up the function. And so when it's saying less rapid areas, it's saying less rapid than the current sinoatrial node, okay? It's not saying the least rapid in the whole heart. That's not what it's referring to. It's saying if the SA node fails, the cells that are the next ones in line for rapidness will take over, but they are less rapid than the original SA node, okay? So each time the heart contracts, it starts with a signal from the SA node. Okay? That is really important to remember. And when that SA node stimulates that signal to contract, that contraction signal spreads from cell to cell to cell. Remember, via our, can I write on here without messing it up? That's always the question. The intercalated discs, and which have our gap junctions. Okay, So the cellular fluid and ions are able to flow between cell to cell, which helps with the spread of the contraction. So it spreads across the atria, okay? So the atria are not just doing this giant instantaneous contraction. The contraction spreads, which is important to remember. And so we're talking about the atria. It is a lot like doing the wave, because you need, let's say we're at a stadium, you're not going to jump up and wave your hands until the person next to you does, and then you do it and get down and the person next to you does it. And so you have to be stimulated, in this case, by the cell right next to you before you can contract and send the signal on to the next one. And so when the atria contract, it literally is like a wave-like contraction, which is perfect for squeezing blood into your ventricles. Okay? And so remember, you have to have the cell next to you be stimulated before you can be stimulated, if you are a heart cell. Okay, so this is really important. All cells are interconnected, but rely on the cell next to it to receive the signal before it can contract. Okay, so we do have an SA node. There also is what we would call an atrioventricular node, or AV node. And so if you had to guess, atrioventricular, so this node is going to be somewhere between the atriums and the ventricles. And so in this case, it's very close to the center of the heart. And I'm just going to pull up a picture here on the internet here so you can see it on the board. Not Moodle. We don't need to talk about grades right now. Heart conduction. Oh, I spelled it wrong. Don't you love the tap there? Just nod. Okay, heart conduction system. All right, so this is great. There's some really good pictures. Let me find one I like. Well, for now, oh my, there's so many good ones. Okay, we'll stick with this nice, easy one. Why is it not coming up? Okay, so this is a nice picture showing the conduction system of the heart. And so the conduction system of the heart is going to require the SA node to be functioning. Remember, that's up in the right atrium. And then that signal from the SA node, if you see this here, so SA node is hot. Why is my writing not working? It won't let me write. Okay, well, you can see the SA node. I'll just point to it up here. Up at the right, kind of upper corner there. So the signal spreads cell by cell through the right atrium. And the signal also is able to affect the left atrium at the same time. And so the atrial contraction is going to be separate from the ventricular contractions. Okay? So the sinoatrial node stimulates both atria to contract in like a wave-like fashion, squeezes that blood into the ventricles. Then what happens next? How do we stimulate the ventricles to contract? We have basically, so very close to the center of the heart. It's not actually in the right atrium. Okay? It's kind of within this center portion, within the connective tissue skeleton that kind of separates the walls. Okay? That signal eventually reaches the atrioventricular node, and notice that you actually have these special little fibers we'll talk more about that travel down. What is this division here between the left and right ventricle? Yeah, the interventricular septum. So the AV node, we have these special fibers called Purkinje fibers that travel down our interventricular septum, and then notice they branch and go up the walls of the right and left atrium. And they even are innervating essentially those papillary muscles which are attached to your chordae tendineae. Okay? So when the AV node receives a signal from the SA node, the signal travels very fast in the ventricular section because we have these special fibers. And so the signal, in this case, is wanting to cause the ventricles to contract together. not wave-like, but like instantaneous. So the contraction, wave-like fashion in the atria is very different than the pretty much instantaneous contraction of the ventricle, thanks to how the AV node is set up, especially with those bundle branches, Purkinje fibers, that branch off. And they're not nerves, but they're nerve-like. So let's go back to our, here we go. So right here, so first of all, it says your AV node has to catch the signal from the SA node as, you know, the cells are contracting and passing that signal on from cell to cell. Remember that. And so it catches that signal, and then very quickly it spreads that signal down through the interventricular septum like you saw, and then up the sides. And there's several terms. Conduction fibers, on the picture we just saw, it called them bundle fibers, and then also often referred to as Purkinje fibers. So there's several terms for that. But those fibers act like nerves so that the signal travels really fast and the ventricles contract all together. Okay? So please remember that how the atria contract, that's wave-like, but how the ventricles contract is almost instantaneous because of the structural difference. We have those bundle branches, those Purkinje fibers that travel down the interventricular septum and up the walls. And so it's super quick. You get an instantaneous type contraction, which is important in how the blood flows in the heart. Okay? And it's based on the structure of the heart itself. So we'll look at that in the next picture. Okay, so remember I told you they're not nerves, but they're nerve-like. And so they function to send signals very quickly, so ventricles contract instantaneously. Okay. So as I said, that signal is, quote, it says caught, but the signal reaches the AV node, which is then able to spread that signal through those bundle branches. Okay, so you have a right bundle branch that goes to your right ventricle, and you have a left bundle branch that goes to your left ventricle, and then the fibers spread up and out like you saw in that picture. And so specifically, those fibers that branch out, those are your Purkinje fibers. So if you want to be super detailed, the bundle branches are within the interventricular septum, if I can write this. And then as it branches out, those are the Purkinje fibers. Okay, so those Purkinje fibers, those nerve-like fibers, spread the signal throughout the muscle. And interesting fact right here, each muscle fiber that receives the signal, they're receiving it at about the same time, as I mentioned before. So if they're receiving it at the same time, we're going to have a different type of contraction. So I know I've said that a bunch, but it's really important to remember the difference in how the heart contracts in the atria versus how it contracts in the ventricle. And so it says right here, so the cells are going to contract at the same time in the ventricles only. So the ventricle has to contract at once to have the optimum flow of blood up and out of your heart. You don't want it to spread or contract in a spreading motion. So if you remember, the ventricles are in a V shape. If it contracted in a spreading motion, the upper portion is going to contract first. If we're following the same idea that it spreads and you have a wave-like contraction like the atria, if we had a wave-like contraction, the upper portion would contract first and then squeeze blood down to the point, and that's not good. It really screws up how the blood is going to flow and reduce it. It would reduce the pumping ability and pressure of the heart because the blood is being pushed in the opposite direction. That's not good. So yeah, it would trap the blood at the apex if it did contract in a spreading motion. We don't want that. So it is V-shaped, and then we have an instantaneous contraction. So if it's instantaneous, it easily, so if I go real slow, it squeezes blood from the apex out which is the most efficient way to squeeze all that blood out of the ventricle, is when you do it instantaneously, it just squeezes it on out instead of spreading it and then trapping the blood. So there's a reason behind why it contracts the way it does. And all this is just what I just told you. And so by having the V shape and contracting at once, you generate the maximum amount of pressure, and that blood then travels out of the heart, especially if we're thinking about the left ventricle. It has to generate enough pressure to have your blood travel the entire systemic circuit. So the heart is designed to work just perfectly. Yeah. So here is a really good summary between the atria and the ventricle. So atria, wave-like. Ventricular is instant, all at once. And notice it even says massive. It is. It's a very strong contraction, which makes sense since you have seen the muscular wall of the heart now, especially on the left side. It's really strong. Okay, this lovely illustration here, we're going to talk about what this means. You do need to know what this means here. So if you, for example, go in and get an echocardiogram, electrocardiogram, where they put the little sensors on you and they're trying to see, they're looking at the conduction system of the heart. They're seeing, are the atria contracting properly? Is there the right amount? Is the signal spreading the way it should? And we can tell a lot of this stuff by looking at these graphs. So this is how it would print out if you were doing this at the doctor's office. You'd have this long little strip, and then the doctor looks at the pattern and of your sinus rhythm in looking at these dips and peaks. And so I'm going to explain those dips and peaks right now. So you need to have an electrocardiogram in order to do this, and it does help to ensure, helps you know if everything is working properly electrically speaking. Like, is the SA node working properly? Is the AV node working properly? Is the heart contracting with the strength that it needs to? So first of all, the P wave right here, notice that it is short and wide. Okay, so it's wide and relatively short. So the P wave represents the spreading contraction of the atrial muscle. So it takes a little longer because it's spreading from cell to cell. So that's why you have this width here. So it's a slower contraction with less force. Okay, so that's why the peak is not very tall. Then you have a little gap. Okay, and this is where the signal is spreading from your SA node to your atrioventricular node, and it's going down the right and left bundle branches, and then stimulates that massive instantaneous contraction of the ventricle, which is the QRS peak right there. Okay, so this whole peak represents the ventricular contraction, and if we note, and it's not as easy to tell in this picture, but the length or the time period of that contraction, how it spreads, is very quick. It's shorter compared to the atrial contraction, and the force is much higher, and the strength of the force you can see by the height of the peak. So QRS represents ventricular contraction, but what does the T represent? Huh? It's going to be the relaxing of the ventricular muscle before the next contraction begins. Again, it is not another atrial contraction. Sorry. Okay, so structures and things you need to know that we just covered for your exam are mentioned here. So it says no, I'm sorry, I'm just going to read this one line because it is important. No sequentially, so in order, the role of each of these structures in the timing of the heartbeat. So know what the SA node does. How does that affect the atria. Know what the atrioventricular node is, where you find it, what does it do, what are those bundle branches, what are the Purkinje fibers. And most importantly, how do those affect how the muscle contracts in either the atria or ventricles. And that is essential for you guys to know the difference in contraction types, or the type of contraction between the two. Okay, so we have a term here, sinus rhythm. It says this is we call the normal beating of the heart the sinus rhythm. Why do we call it the sinus rhythm? I'm going to back up for just a second and point out that this little graph here, the representation of the strength of contractions and the rest periods, that's the sinus rhythm. Okay, this is how we are able to read it with an electrocardiogram, how we're able to kind of take apart what's going on with the atria and the ventricles, etc. So we call that pattern of the atria contracting and then the ventricles the sinus rhythm. But why do we call it the sinus rhythm? It's because it all starts with the SA node. That's why we call it the sinus rhythm, because it begins at the sinoatrial node. Okay? Because that's where the whole thing begins. Oh my goodness, really? Okay. Let me just check if there's any... No, wait. This is the last slide? I think so. Hold on, I just want to double check and make sure I think it is. Okay, so we finished the whole chapter exam on Friday. Study guides are up and available, and office hours for myself and TAs are available. And let me know if you have questions. Yes, sir?
Is there any coloring due?
No. No homework at all is due on Friday.